Now that we've gotten some of our introductory material out of the way, we move into a topic that, I'll be honest with you, some of you guys are going to absolutely despise. However, it is important for us to get this topic under our belt. If we can get even a rudimentary understanding of some of the basic chemical topics, the other topics that we have to discuss moving forward are going to make that much more sense. We can't discuss physiology without using terms like ions, different types of bonding. We can't talk about diffusion or movement of molecules into and out of a cell without getting into some of those topics. So rather than jump straight into those topics, let's go back and do an introduction of some of the more basic chemical topics that we're going to need. You're going to have to get an understanding for basic chemistry. Atomic and ionic structures, different types of bonding like covalent, ionic bonding in particular. And we're going to introduce you to the concept of pH, acids versus bases. But admittedly, much of this topic is going to be covered further down the road. First of all, let's look at atomic structure. It's likely you've already seen this at some point, whether you have put those memories in the recesses of your mind or not. But I'm sure this has come up at some point. An atom is going to be made up of a nucleus containing positively charged protons and neutrally charged neutrons. Surrounding the nucleus, and I hesitate to say this is what it actually is. There's more detailed models of this. This is not exactly how it behaves. We're going to keep this at the level that we need to. Nucleus with the protons and the neutrons. Surrounding the nucleus are going to be our third subatomic particle, the electron, orbiting the nucleus in layers. Each of these layers we're going to call an orbital. The outermost orbital we're going to call the valence orbital. So protons in the nucleus, positively charged, electrons orbiting around the outside in layers that are negatively charged. And when we consider an element, like you would see on our periodic table, generally speaking, these are going to be atoms that are neutrally charged, meaning the number of electrons is going to be equal to the number of protons. Done. Where we get ions from, and this is going to be the entity that we're going to be referring to the most, that students seem to really not grasp what it even is. In order to go from an atom to an ion, all we simply do is manipulate the number of electrons. What makes an atom want to change its number of electrons? And that's really straightforward. An atom is always going to strive to fill its valence level orbital. Remember, the valence level was the outermost ring. It's going to strive to fill that level up with as many electrons as can fit in there. Now, we can't just pack them in there indefinitely. They can only hold so many. The first level can hold as many as 2. Only 2 electrons can fit in that inner first layer. When you get 2 filling up that inner layer and you want to get a bigger atom, move up the periodic table, if you want to put a third electron into this atom, now you got to put that third one in the next layer, a second layer. A second orbital layer of an atom can hold as many as 8. If we want to get a bigger atom again, we would need to add a third layer. The third layer can hold 8. Now, as you can imagine, we've got an awful lot of elements on the periodic table. As we get bigger and bigger and bigger, we're going to be adding more and more layers. Now, beyond the third layer, things get extremely funky. 2 in the first layer, 8 in each of the next 2 layers. Once you start getting into a 4th layer and fill that, then you can go back and put some more in the 3rd and grow that way. But we won't need to get into any atoms that even reach that size. We won't get into any atoms in our discussion that go beyond 3 orbital layers in size. So 2 in the first, 8 in the second, 8 in the third. That's the maximum capacity of each of those layers. An atom is always going to strive to have a full outer layer. So let's look at chlorine. Chlorine is an element that we see in the periodic table over here. Chlorine has 3 orbital layers. We see the first one, it's been filled up, it's got 2. We see the second layer, 2, 4, 6, 8. 8's its capacity. We go into the third layer now, its valence level. 2, 4, 6, 7. A chlorine atom has 7 electrons in its valence level. How many would make it full? That third layer can hold as many as 8. It's 1 short. So it's going to have an affinity for an additional electron. It's going to attract 1 more electron to fill up its outer level. That's where it's most happy. That's where it's most stable. So we take on a single electron, plop it in that valence level orbital, and now we've got the full set. 2, 8, and 8. But by doing so, we've offset the electrical balance. When it only had 7 electrons in its orbital, the number of electrons it had was equal to the number of protons it had. But now that we add an extra electron, we've got 1 more electron, 1 more negative charge than we have positive charges in the nucleus. So as a whole, we give this entire chlorine ion a negative charge. So an ion is an atom that has gained or lost electrons, therefore offsetting that balance. It's always going to be a charged particle. Over here we have sodium. Sodium is going to be found here on the periodic table. Now let's analyze sodium in the same way we analyze chloride. We have 1 orbital level here, containing 2. It's full. We move to the second, 2, 4, 6, 8. That's as many as it can hold. We move out to the third, and we've got 1. We've got a single valence electron. Now, we always want to make sure that valence is full. The outermost orbital level, we want it to be full. We could gain 7 and fill up this third one. Getting in 7 is a bit of a job. It would be easier for us to simply take that lone electron and get rid of it. We get rid of that single electron. That gets rid of this entire orbital level, leaving behind a new valence level, which already contains 8. We lost 1 electron, meaning we lost a single negative charge from the structure. It offsets the balance. Now we've got more protons, positive charges, than we have electrons, negative charges. Therefore, the sodium ion takes on an overall positive charge. Anything carrying a charge is always going to be water soluble. That's important. And an imbalance in those protons to electrons is always going to be considered an ion. You'll hear a couple of terms like anions and cations. Anions refer to negative ions, cations refer to positive ions. So now let's look at 3 different ways in which we can bond these elements together. Because if we look at a periodic table, we're only talking about one single entity. But we want to have ways in which we can attach these entities together to form different compounds. The first type we have is ionic. That's where I like to start. Ionic bonding should look the most straightforward to you. What would you suppose would happen if we got one negatively charged ion and one positively charged ion? What would they do to each other? Those opposite charges are going to attract. That's ionic bonding. We take two oppositely charged ions, we bring them into close association with each other. Those opposite charges attract, and we get a bonding effect between those two ions. Now, if we take something that's ionically bonded like that, and we put it in water, they can separate. So both ions can kind of free flow through the solution. They disassociate. Now, I want to take that principle, that positive, negative attraction, and use that paradigm to move into our next type of bonding, covalent bonding. Now, these would be the type of bondings that we would see. If we draw them in a textbook or on a piece of paper, we would see something, X bonded to Y. And we would draw that line in there, and it implies that we have some sort of hard fixation between the two, and not just a mutual attraction. You and your partner may be bonded by mutual attraction. You go places together. You're in close association with one another. This, under that analogy, would be both you handcuffed together. It implies a hard bond. However, I don't want us to look at it as if it's a hard bond like that. I want to take what we visualize already with our ionic bonding and translate that over into covalent bonding, because it's not as different as it first appears. Let's look at this diagram on the right. This is carbon dioxide. Remember what I said about ions. They form that way because the atoms want to always fill up their outer levels. Always. Covalent bonding essentially accomplishes the exact same thing. Here's oxygen. 2 orbital levels, 2 in the first one. Wonderful, it's full. Count the red ones now that we see in the second. 1, 2, 3, 4, 5, 6. How many electrons can fit in that orbital? 8. We have 6. Same goes for the oxygen on the other side. Now if we look at carbon, 2 in the first layer, perfect. 1, 2, 3, 4 in the second. There's 4 electrons in that layer, of which the capacity is going to be 8. All of these atoms are going to want to fill up their outer levels, just like we saw with the formation of an ion. However, unlike an ion, where we have the atom gain an electron, we don't accomplish that same feat in the same way for covalent bonding. We share. The carbon is going to say, I share some of yours, you share some of mine. So it appears as if we have filled our valence levels. So if the oxygen borrows 2 atoms, or 2 electrons from the carbon, now we count its original 6 plus sharing 2 from the carbon, we can count 8 in its outer level. Do the same thing with this particular oxygen. Likewise, now when we count the carbons with all of that sharing, 1, 2, 3, 4, 1, 2, 3, 4, and you may see these electrons now line themselves around all valence levels from those 3 atoms. but it gives the impression that each atom has filled their valence level, even though we didn't add or lose electrons in order to do so. This still creates a very stable, happy arrangement that these atoms are perfectly comfortable in. Now, where I want to reinforce that we want to always keep our ionic bonding in mind is now when we move on to a concept within these covalent bonds called electronegativity. We talked about sharing. I don't know how many people out there interact with other people often, if you have kids especially. Very often when things are being shared, they're very often not shared equally. I've got a 12-year-old boy and an 11-year-old little girl, and you've never seen two people scheme to try to get an advantage over the other. I swear negotiations don't take place like this at a UN meeting when they're trying to divide up pieces of pizza. So when we see sharing occur between two atoms, we don't expect that sharing to always be equal. Some atoms, because of the size of their nucleus, can attract electrons closer to them than the other atom in the pair. This is water. Water has a much larger nucleus than hydrogen. So any shared electrons won't reside right in between the two, but closer to the oxygen. There's an imbalanced, unfair sharing. The end result is still the same. They still wind up counting towards the filled valence levels. They just don't sit right at the midline. The relative attractiveness for those shared electrons is referred to as electronegativity. And because of that unpaired sharing, now we can apply vectors in the direction of where the electrons more accurately reside. So for this particular covalent bond that we have here, the electrons are closer to the oxygen. Therefore, we have an overall vector in that direction. We get the same thing in the other one. Whenever we can apply those sorts of vectors to a covalent bond, we refer to it as a polar covalent bond. Water, because it's angled at that V-like shape, and we have two polar vectors, we have an overall polar vector observed through that water molecule, making it a polar molecule. If we look at our carbon dioxide, it hardly mattered because of the way they line up. Even if we do have any vectors within their individual bonds, they're going to be canceling each other out. This results in a non-polar covalent molecule. Now, why does this matter? I said before, whenever you see anything with a charge, those are always going to be water soluble. That makes a difference for later on. Polar molecules are always going to be considered water soluble, whereas non-polar molecules are going to be considered non-water soluble or hydrophobic. This matters a great deal once we start talking about how different types of molecules or ions get into and out of a cell, based on polar, ionic, or nonpolar. We will see different means of transport used for ionic and nonpolar molecules. We will see different modes of transport used for large molecules versus small molecules. So getting some sort of perspective on what constitutes those particular characteristics is going to help you once we move into those sections. Hydrogen bonding is going to be the result of multiple molecules having this strong electronegativity. If we have molecules that have a composition of hydrogen along with oxygen, nitrogen, or fluorine, we will see hydrogen bonding occur throughout a solution. So when we look at three different water molecules, knowing that we've got strong electronegativity and polar characteristics to that particular water molecule, we can essentially line up those and have a positive pole and a negative pole. Now that we've got this as a covalently bonded molecule, applying once again what we know about our ions, what keeps this positive pole from bonding to the negative pole on a neighboring water molecule? That relative attraction is called a hydrogen bond. This is going to apply to certain things like surface tension of water, anchoring those independent water molecules together by an electrically attractive force. The next thing we move into is acids versus bases. And this is the topic that won't even really get elaborated on much until you move into A and P2. But we just finished up talking about hydrogen ions. Hydrogen ions are what plays a role in the relative acidity of a solution. In order for us to measure the acidity of a solution, we have the equation of the pH log. The pH is a scale of relative acidity, with the low numbers, 0, 1, 2, 3, 4, being more acidic, the higher numbers, 10, 11, 12, 13, 14, being more basic, 7 being neutral. Distilled water would be 7 on the pH scale. The only thing that matters when we talk about pH is the concentration of the hydrogen ions. pH is equal to the inverse log of the concentration of hydrogen. As the concentration of hydrogen goes up, it becomes more acidic, which makes the pH number get lower. Normally, pH of blood is anywhere between 7.35 and 7.45. Now, those are, I mean, in the big scheme of things, those are small numbers. Those are smaller numbers than we dealt with when we were talking about body temperature, blood sugar. But even though we're talking about 1/10 of a point, this is a remarkably narrow range. Because remember, in order to get to that range, we use the inverse log to get there. Anybody who's ever done a chemistry experiment in a class called a titration knows how narrow that range is. A titration lab usually involves an acid of known pH, known concentration, and above it, in a long tubular flask, tube with a valve at the bottom called a burette, you put a base with an unknown concentration. And through the mathematical formulas that you would need, if you add enough base to the known acid to get a pH reading of exactly 7, now you can calculate the pH of your unknown base. So you add a little bit of base, and you're measuring the pH all the time, and it hardly changes. You add a bit more base, pH hardly changes. The closer you get to 7, it gets a little sticky, and you will get to a point where you're doing like 1 drop at a time, a half a drop. Ever do a half a drop? You get just the drop hanging off the bottom of the burette, and you close the valve, and you take some distilled water and rinse off the tip of the burette to get that half a drop into the acid. It's usually about that point where you get nervous and you open the valve a little bit too much, a bunch of base falls in, and you completely ruin your experiment. You got to start over. But remarkably narrow range. The pH of blood has to be maintained within this range for the viability of the blood cells, for the integrity of the hormones that are circulating, the enzymes that are circulating. So we want to maintain pH in this narrow range. And we do so with 3 different mechanisms. Kidneys. If you've got too much hydrogen ions throwing off your pH, you urinate them out. If you've got too few hydrogen ions making your blood more basic, you retain more. The buffer system is the chemical system that helps stabilize. And it's going to be, or at least the one we're going to discuss, is going to be directly related to your respiratory system. So to keep things in context right now, I want you to know what an acid versus a base is and what a buffer system is. And that's where we're going to call it on that topic. An acid is any chemical that releases free hydrogen ions into a solution. If you have a molecule, you put it into water, and that molecule gives off free hydrogen ions, that's an acid. We need those free hydrogen ions in order to calculate pH. A base is going to be any substance that consumes those free hydrogen ions, reducing the number of free ones we have in a solution, therefore elevating our pH number. A buffer system stabilizes the pH of a solution. We have such a thing as strong acids and weak acids, and strong bases and weak bases. Don't fall into the trap of thinking an acid is stronger than a base. They are equally strong on opposite sides of our pH scale. What a buffer system will do is it will stabilize your solution. If we add strong acid to that same solution, the buffered chemical system takes that strong acid and converts it into a weak acid, one with lesser pH effect. If we add a strong base, it takes that strong base and converts it into a weaker base, having less of a pH impact. But it's important to realize that a buffer system is a chemical system. We have to have a weak acid and we have to have a weak base in the same system. The one I want you to know is our carbonic acid sodium bicarbonate buffer system. This is what happens in the blood. We have sodium bicarbonate, a weak base, and we have carbonic acid, a weak acid. So if we add strong acid, it reacts with the weak base, producing a weak acid. So we've taken the strong acid and converting it into a weaker acid. But that's only half of what a buffer does. It also has to compensate for the bases. Look at the similarities between these two equations. Sodium bicarbonate, sodium bicarbonate. Carbonic acid, carbonic acid. If we add strong base to the same system, now the strong base reacts with the weak acid, producing a weaker base. So we have these two chemicals in the solution at all times, and it doesn't matter if we add some acid or we add some base, that chemical system helps compensate. Now, where we link this into respiratory, is all about our carbonic acid. We said earlier that an acid has to be a chemical that when added to water gives off free hydrogen ions. This is called carbonic acid, so it had better do that, right? And it does. Carbonic acid, H2CO3. It's going to give off hydrogen and carbonate. There is your acidic breakdown. However, carbonic acid doesn't only break down like such, it also breaks down into water and carbon dioxide. Now, if you notice, we drew a couple of fancy arrows in there. Arrows that are going in two different directions. In chemistry, we use those arrows when we refer to something called an equilibrium. It implies that this reaction can take place in both directions. Carbonic acid can break down into hydrogen and carbonate just as much as carbonate and hydrogen ions can form carbonic acid. Carbonic acid can break down into water and carbon dioxide just as readily as it can form again back into carbonic acid. The importance comes into we have the ability to manipulate carbon dioxide levels using our respiratory system. If we disrupt a chemical equilibrium, the result is going to be the establishment of a brand new chemical equilibrium. So, for example, how can we manipulate carbon dioxide? What happens to my carbon dioxide levels if I hold my breath? If I'm not breathing out carbon dioxide, levels in the blood increase. If we increase carbon dioxide, we are going to see an increase in carbonic acid, causing the pH of the blood to go down. If I hyperventilate, if I've got any number of anxiety issues and I hyperventilate, have a panic attack, I'm breathing in and out very rapidly. Every time I breathe out, I expel excess carbon dioxide. That's going to deplete my blood of carbon dioxide levels. I disrupt the equilibrium by expelling too much carbon dioxide. We establish a brand new equilibrium, which means we have less carbonic acid present. We will always see a direct relationship between the concentration of carbon dioxide and pH of blood because of that particular breakdown. Now again, as far as pH goes, know what acids and bases are, know what that pH scale means, know what a buffer system is. We will not be expecting you to know the respiratory system at this point. You will get back into this again when you do respiratory in A and P2. But we see any number of things that can throw off blood pH as a result. You can have a respiratory acidosis when you breathe too little, like I mentioned before. Now, hypoventilation is not always the simplistic example that I gave of simply just holding your breath. You also get effective hypoventilation for various respiratory ailments like COPD, asthma, and emphysema. Maybe you're breathing at the right rate, but not exchanging gases like you're supposed to. Brainstem injuries can throw off respiration rates. When you do respiration, you'll talk about the different control centers there for respiration. You can have a respiratory alkalosis. Now, a good example there is going to be that hyperventilation. Right? People hyperventilate. What do? They give them a paper bag to breathe into. Now, you're breathing in and out too quickly, but as you breathe into the paper bag, you're breathing back in some of that carbon dioxide that you expelled out. So that paper bag helps stabilize the carbon dioxide loss. High altitude. You go from Lexington, Kentucky to Denver, Colorado. Now, in order to get the oxygen you need, you got to breathe twice as fast. If you need to breathe in twice as fast to get the oxygen you need, unfortunately, you need to breathe out twice as fast as well, expelling excess carbon dioxide. You'll see when you do respiratory that gas exchange is not a trade. The movement of oxygen across the lining of the lungs is only dependent on the concentration gradient of oxygen. Carbon dioxide moves in the opposite direction based on its concentration gradient. It's not simply, you go that way, I'll go this way. One for one. They move independently based on their own independent driving forces. Now, unlike the respiratory ailments, what about if you didn't have a problem with your carbon dioxide levels through breathing, but you were simply producing more acid? Some of the byproducts of metabolism increase hydrogen ions. You can get a metabolic acidosis, renal disease, starvation, excessive diarrhea, causing dehydration. Atkins diet puts you in the state of ketosis, which can increase acid levels. Strenuous exercise, you produce more lactic acid. So rather than manipulating the CO2 levels to change pH, you're dumping more free hydrogen ions into the mix, throwing off the equilibrium. Now, as a result, you may want to change your respiration rate based on your metabolism. You can also get a metabolic alkalosis. Think about the stomach acidic contents. If you vomit those up, they have to be replaced, depleting blood acid levels. Now, where we go to next is a continuation of our chemical discussion, but it really moves us more into the biochemistry. We're going to look at the three types of macromolecules: carbs, lipids, and proteins. We'll look at the structure of each, but more importantly, we're going to look at how our body breaks those down so that we can convert the energy in those molecules into energy in the form of a high energy molecule known as ATP. So we'll be looking at the metabolism of carbs, lipids, and proteins so we can produce our body's universal unit of energy as ATP. ATP, adenosine triphosphate. And the last thing I want to touch on here with this lecture is going to be energy. Where in a molecule do we store energy? That seems to be an abstract thought that pops up a lot. Energy is always stored in the bonds. It should make logical sense that if we take a bunch of independent atoms and we want to make them into one bigger molecule and bond those atoms together, that would take energy input. That energy is stored in the bonds of that molecule. And when we break the bonds again, we can release that energy. ATP, adenosine triphosphate. This particular bond is extremely high energy. So you will see us take those macromolecules, create molecules of ATP. We break that bond, releasing the energy and leaving us behind with ADP plus this lone phosphate group. But energy is always going to be stored in the bonds. So again, know all relevant terminology. You should know basic atomic structure: ions, atoms, molecules. You should be familiar with our types of bonding, particularly covalent and ionic. And where we take that information and make it directly relevant is by determining its water solubility. Ions and polar molecules tend to be water soluble, hydrophilic. Non-polar molecules tend to be insoluble, more hydrophobic. Those two characteristics will determine how those molecules and ions are able to get moved into and out of a cell. You should know what pH means. You should know what an acid versus a base is, the pH calculation. And you should know what a buffer is. Even though we're not expecting you to understand the full feedback mechanism and how it ties into respiration, you should know basically what a buffered solution happens to be. And the one we use for the blood is the carbonic acid sodium bicarbonate buffer.